Okay, so today's webinar speakers are John Vasilic and Austin Smith. The first half of the webinar is presented by John and the second part by Austin. John Vasilic has worked at BMS for 29 years in the area of analytical method development and support for the process development and production arenas. His group focuses on non-invasive green spectroscopic applications and serves as a representative on BMS sustainability team. Austin Smith has worked for the past 5 years as a process chemist at Amgen. He's a member of the Drug Substance Technologies Organization, where his work focuses on commercial process development in support of Amgen's small molecule pipeline. He is also the Amgen Green Chemistry team lead. Now, I think, John, are you ready to get started?
Yes, I am. Thank you very much for the introduction, Ingrid. For the first slide, I'd like to begin with thanking everyone who is dialed in. It's either good morning, good afternoon, or good evening, whatever the case may be. The first set of slides will be really an introduction into the concept of green chemistry. If you're familiar with it, that's great. If you're not, take a careful look at this. So we're going to go over the ins and outs of green chemistry, and I'd like to begin with really what is it, a definition. And I'm actually going to read this straight out because I think it's very appropriate and very well written. Innovative science aimed at design, development, and implementation of chemical products, reactions and processes that minimize hazardous substances and are inherently safe. They reduce waste. You want to reduce the environmental footprint while improving efficiency and economics. I want to point out here that obviously, since we are scientists, we always like to be challenged. That's why we got into science. And as Tucker put this back in 2006, a privileged opportunity for innovation. Once you're challenged with green chemistry and looking at sustainability, it enables you to hopefully rise to the top and meet challenges, to come up and be creative. Some people are mainstream, some like to think outside the box, and some like to think way, way outside the box. And that's what drives the innovation and why we went into science to begin with. Now, green chemistry is really synonymous to sustainability. Sustainability is just the ability to maintain a certain ratio or certain rate or level. In the idea of ecology, it's really to avoid the depletion of natural resources in order to maintain an ecological balance. And in this particular case, if you look at the examples here, what is outlined? In industry, well, there's an economic capital. The less waste we produce, the less we have to pay for disposal, the less costly the material is. If you can run something in an aqueous condition, it drives the cost down. So there's an economic capital there. This also affects the environment, the natural capital. You don't want to ruin the environment, obviously. Personally, I don't know anybody that says to me, hey, I'm going hiking this weekend. I think I'm going to go hike a Superfund site. We want to keep the environment clean and natural. And of course, that society, that's where the impact is. I'm going to go hiking in a nice place. I don't want to live near a Superfund site. There's the human value in keeping what we already have available in the world today. So these things are really intertwined. And as you reduce waste, you end up reducing cost, not only in the front end, but on the back end when you do have to clean up. So this is why it really is important to try to think of green chemistry as a sustainability goal for the future. Now, back in 1990 came the Pollution Prevention Act, and this was really a change in mindset, where it wasn't a matter of addressing just the pollution we had created. It's looking at the source, and the source reduction was a goal. So can you substitute raw materials that are less toxic? Can you develop processes and utilize equipment that will decrease the amount of waste that's generated. And of course, even much more mundane items that really rose to the top, such as inventory control, just-in-time synthesis, continuous flow chemistry. You wouldn't think of it as having a big impact, but it really does. Even housekeeping and maintenance, making sure your equipment is in tip-top shape so it runs efficiently, much like you want your car to run efficiently. Now, this does tie in with green chemistry's hierarchy, with EPA's hierarchy, and they're really well harmonized. If you think about it, you want less hazardous items to reach humans, to reach the environment. We have some prime examples today where can you reuse or recycle chemicals and not have to worry about waste disposal and the cost and the danger that goes along with it. So these are the items that we're trying to relay that are most important to reduce chemical waste. Now, here's a couple of examples, and they're varying examples, but some of them hopefully are eye-opening. Computer chips were originally made that required a high amount of organic solvents, a lot of energy input, water, and so on. The advancements, well, over time, they came up with a better way. Supercritical fluid CO2 really reduced the amount of water and solvents and energy that was used to make computer chips and the circuit boards. And the boards are now made of recycled chicken feathers. What normally would have gone to waste can now be reincorporated into making the boards. So it's a really nice example of a green approach. On the drug side, sitagliptin. Now, that's a unique compound. You may know it as Januvia for type 2 diabetes. They used some metal catalysis when it was first made. What Merck and Codexis did is reinvent it, take a look at it, and now have an aqueous-based enzyme process that replaces the metal catalysis. And so it's green all around. It helps because now you're dealing with an aqueous system. You don't have to worry about exposure to metal catalysis. You don't have to do additional assays to make sure you removed it, and maybe had to do extra steps to drive that down to near zero levels of metal in your product. It's a win-win situation. If you think about plastic bags, the plastic bags now are biodegradable, and they've added natural additives to it. They're actually stronger as long as they don't make them too thin, and printable, much better than the petroleum-based plastic bags from years ago. And I remember also as a kid painting my bedroom and having to sleep someplace else in another room for really 2 days because of the oil-based paint let off so many VOCs. So the fumes were just too much to handle. Now, water-based paints are very, very prevalent, and they've modified them so that they can use, for example, micronized plastics to help build some durability in the paint itself and washability. So they're looking at how to improve what we already have and make them less toxic. So these are chemistry related, but it shows you a wide range of advantages. Now, if we go into the history, and we're looking back about 60 years ago when R.B. Woodward introduced the concept of synthetic design. This was a smart chemistry approach to take a look at the entire synthesis and how to reevaluate how to make something. Probably a big impact came from Rachel Carson back in 1962. She was a marine biologist who, if you're familiar with Maine, hung out in the Down East area. And in The Sea Around Us, which she wrote in the mid-50s, she began to notice that some of the estuaries were becoming poisoned by pollution runoff, pesticides, things that were used in the potato farms and so on. Found trace metals, things washing ashore in the estuaries that were affecting the fish breeding population. So her book, Silent Spring, really opened up people's eyes to look at the impact of pesticides, of pollution. And of course, we've gone on and other people have contributed. E.J. Corey won his Nobel Prize on retrosynthesis. And then the concept there is take your target and work backwards. Simpler precursors. The goal, structural simplification. Barry Trost came up with the atom economy or atom efficiency. And that's the efficiency of a process so that in terms of all atoms involved and the desired products are the same. So in an ideal world or an ideal synthesis, no atom is wasted. The anti to that would be like a Wittig reaction, where you're using a high mass phosphorus reagent that then gets disposed of and goes into hazardous waste. So that is a negative impact on your atom economy or atom efficiency. Now, my colleague Austin is going to be talking about some of the other important items that were developed, specifically the E factor, for example. So that will be coming up more, but this is going to tell you how you take a look at the chemicals you have and what kind of value you can assign to its level of toxicity, handling, waste, and so on. In the mid-1990s, a presidential Green Challenge by President Clinton at the time really tried to reinvent environmental regulations and promote pollution prevention and industrial ecology. And he wanted a new industrial partnership. This was really cool because it brought in industries, organizations, groups, and individuals can compete for this. So this was really a big step forward. The Green Chemistry Institute was launched in 1997, and a few years later was incorporated into the American Chemical Society. And that was followed by Anastas and Warner's Green Chemistry Theory and Practice, the 12 principles of green chemistry. These still are extremely important, very well quoted, and really the backbone of the green chemistry approach. I'm going to have some other slides to go over the principles in just a moment. Also in 1999 marked the launch of the Green Chemistry Journal by the Royal Society of Chemistry. I highly recommend that people pull it up. There's some really innovative science that's going on there, and it is a pleasure to read. Anastas and Zimmerman took another step forward and looked at the 12 principles of green engineering. Again, expanding the concept because chemical engineering, it all comes into play in terms of green chemistry. The roundtable, the GCI roundtable, was formulated with pharmaceutical industries in 2005. And 5 years later, the IQ Consortium on Green Chemistry was chartered. Soon to follow that was the process mass intensity. So you can actually look at your process and come up with metrics and rate the system. And again, Austin's going to be talking about that shortly, along with green aspiration level, important factors that led to the incorporation of the green scorecard, because you have to be able to measure in order to understand what you're doing and to make improvements. Now, those 12 principles of green chemistry, many of them are obvious. Prevent waste, safer chemicals. You want to minimize the potential for accidents. So obviously you want less explosive items. You want to maximize atom economy, which we've already discussed briefly. At the same time, catalysts. What kind of catalyst do you use? Catalysts, you want to make them less toxic. Can you use less toxic catalysts or change what you're doing? All of these are really important, and what's near and dear to my heart is analyze in real time in order to prevent pollution. This is why we incorporate a lot of spectroscopy, PAT. If you just need to wash a cake of API and remove some material that should not be there or residue from your reaction step, it's nice to be able to do it in line quickly so that you know that, well, I'm not going to necessarily pump in 6 volumes of solvent. I can see that at 4.3 volumes, I've already removed all the material, so I'll stop at 4.5 volumes. That's a savings right there. It's an economic saving and environment saving. Now, the impact This is something you can always discuss. People have different opinions on it. You want to avoid chemical derivatives. You want to maximize that atom economy. You want to prevent waste. What the impacts are somewhat challenging because when I look at something that says minimize the potential for accidents, that's hazard, and it's pretty straightforward. And you want to decrease toxicity. At the same time, if I can cut down on the catalyst, especially toxic catalysts, I've changed the toxicity level. I've decreased the hazard material. There's less waste, and I may be able to cut down on my materials that I'm using. Using safer solvents, it's a win-win situation. It does have a positive impact on the environment. Some people say it'll just positive impact on your materials that are going in. All of those add to green chemistry. But this gives you a nice checklist. Now, what I have here is a couple of examples that are very nicely done. The very first one is a solvent-free synthesis of paramethane diester derivatives. It's derived from citronellol, which is a natural product from a plant. And here they're able to use a polymer-supported scandium triflate catalyst. If you're able to immobilize a catalyst and you don't have to worry about removing and going through extensive process later, that helps considerably. The silica-supported sulfonic acids in a solvent-free system is another nice example where you're avoiding solvents, you're using a heterogeneous catalyst, and you're able to get a high selectivity in your product. A nice example. The last example here, the isosorbidide and dimethyl carbonate, I call this a nice green match. It's really taking a sugar, disorbitol, and using DMC, the dimethyl carbonate, with a small amount of base to make anhydrous sugars, isosorbide. And you can carry that reaction further to actually make isosorbide-based polycarbonates and dimethyl isosorbide, which is really a high-boiling, non-toxic solvent. So there's a lot of uses that come out of this study. This is something that I highly recommend that people look into. This is the creativity that sometimes we need in green chemistry. I happen to like this next example. Put this separate. This is for the synthesis of artemisinin. It's an anti-malarial drug. You can make this in a primary country. You can also do this synthesis in a third world country. Now, Martin Plyakoff and Mike George from University of Nottingham, working with colleagues from University of Bristol and Southampton, along with, I think it's up to 24 industrial companies that are joined this group, are looking at ways of doing continuous flow photochemistry in a small to even large scale up. Here you've got a continuous flow reactor. As it's spinning, you've got air, actual room air that's being pulled into the system. So it's a photochemical reaction that uses oxygen just from air in order to do this oxygenation. Really nice stuff, really nice chemistry. I highly recommend everyone take note of this article and look at it. So then, when is the right time to apply green chemistry principles? I mean, what hits me right off bat is the annual disposal cost that drives up the cost of drugs, that drives up the cost of things that we manufacture, various types of chemicals. So as a result, we spend a lot of money, billions and billions of dollars, in order to treat hazardous waste. What I want to draw your attention to is BPT. Obviously, the T is for toxic. There's not much you could say about that. But when you think about the chemistry that you are designing, you don't want to have something that breaks down very slowly in the environment or even in your vessels. This is something you want to take control of. And what's been in the news more so over the last 6 to 10 years is the bioaccumulative effect, where toxic compounds can linger in a living organism, in the plants that we consume, in vegetables, in fruit. Likewise, disposing of drugs, for example? Do they go down the toilet, eventually find their way into the water system? Those animals, I'll put it, such as cows that consume a lot of water, and are we finding some of those remnants in milk, pesticides and so on? All of those come into play. So this bioaccumulative effect is not the way we want to go. So these are some of the items that when you look at green chemistry and you use the tools that are available, you look at what you can do. Can you actually break something down prior to disposal from toxic to less toxic components, which might be a very economical and wise step, but it's something that we have to train ourselves to look at. And of course, when do we apply green chemistry principles? I used to feel that once you have your proof of concept, your synthesis lock or pre-lock synthesis for making, for example, a pharmaceutical is the ideal time. The more I think about it, it should be driven to earlier during the development cycle, partly because the people, even though they're generating small amounts of waste in drug discovery and leading up to phase one, if you already have the mindset of looking at what is less toxic and being creative without sacrificing speed and agility and delivering the compounds, but something that you, at least you have the tools in front of you, you may be able to make some decisions that help the later stages of development. And this is something I like to strongly stress. This is something you should consider in the future for sustainability. With that, I'm going to pass this over to Austin, who will now take control and carry on the second half of this talk.
Thanks, John. I think that was a really nice introduction as to the importance of green chemistry and when to apply green chemistry principles. The second half of the talk is going to focus on how to best measure green chemistry and waste in process development, limitations of some of these metrics, and how we could potentially improve them. And so to start, we ask ourselves, what makes a good metric system? It's important that your green metrics are clear and simple, easily defined, easy to calculate. By definition, measurable, and the last, very important, you want your metric system to be objective. And this is actually a point of contention in green chemistry, especially as it pertains to metrics, and when one should begin starting to count process waste in development. The results of a good, reliable, and robust metric system are an easy evaluation of a process, especially in being able to measure improvements in a process, increase the understanding of the impact that one has made during process development, and then thus be able to communicate out simply into broad audiences the improvements that teams have made. And so I have a really great quote at the bottom of this slide by a famous management consultant and author, Peter Drucker, who said, you can't manage what you don't measure. And perhaps a more famous quote would be by Lord Kelvin, who once said, to measure is to know. And so I think a lot of green chemistry can be rather intrinsic to chemists, but it's very important that we capture improvements, we capture waste in process development as we strive for commercially robust and efficient processes. Okay, so there are many different types of process metrics that have been invented in the pharmaceutical industry over the years. It's important to remember that each one of these has benefits and limitations or disadvantages when it comes to other areas of assessing a process. And so I've highlighted here in the columns waste, yield, consideration for stoichiometry, consideration for solvents used and amounts of solvents used, as well as water waste. So there are pluses or minuses, checks and balances with each one of these metrics. I've highlighted a few in particular that I'm going to talk about in some detail today. That's chemical yield, atom economy, environmental impact factor or E factor, and process mass intensity or PMI. Okay, and so just a quick outline here. So yield obviously is the most simple of the green chemistry metrics. Atom economy was previously discussed by John in as an introduction, and then E factor and process mass intensity. I'll show how these two metrics are intrinsically linked. So what I want to just briefly mention before we get into some of these process metrics is life cycle assessment. And so life cycle assessment is undoubtedly the most comprehensive green metric that we have. It is an end-to-end or cradle-to-grave approach, capturing a full range of environmental effects assigned to a particular product of interest. And so this is an attempt to capture the environmental impact, not just with waste, but in energy use, toxicity, safety risks and hazards, and overall environmental impact, starting from raws, making their way to manufacturing, distribution, use, and then eventually their disposal. So more or less, this is out of scope for today's talk. Today's focus is in particular on the manufacturing section of an overall life cycle of a product, that being the manufacturing process, its efficiency, and then the waste that is produced. The important point here is that life cycle assessment is more than just a process metric. It is a true cradle-to-grave approach to assessing environmental impact for a product. Okay, I won't spend too much time on this first metric because I think as chemists, this is obviously very well known and understood, but chemical yield is the most simple and straightforward measure of the efficiency of a process or of a chemical reaction. It is simply put, the moles of product produced divided by the moles of limiting reagent multiplied by 100. And so in other words, how much of a limiting reagent was converted to product over a given chemical reaction or a series of chemical steps. And in a general sense, the higher the yield of a chemical reaction, the lower the amount of waste produced. However, I think this one's pretty clear in that yield is limited as an overall process metric, and I'll get into that in a few slides. Okay, so moving from yield to atom economy. Atom economy was introduced by Professor Barry Trost at Stanford in 1991, and it aims at addressing waste in a more effective way than chemical yield. And so simply put, atom economy is a measurement for the number of atoms or percentage of atoms wasted when making a particular product. And Professor Trost has introduced this metric back in 1991, and in 1998 actually received the Presidential Green Chemistry Challenge Award for this metric in particular. And so it is also rather simple to calculate. It is nothing more than the molecular weight of the product divided by the sum of the molecular weight of all raw materials and the sum of molecular weight of all reagents used, taking stoichiometry into account. And so this is important because it captures efficiency in a different way than yield. And I think maybe the best example would be what I've shown here on this slide, which is the first commercial process to make phenol, in which a process can have 100% chemical yield, as in this case, but actually produced more mass waste than product. And so atom economy essentially focuses on emphasizing a disadvantage of high molecular weight protecting groups, high molecular weight and the use of auxiliaries, as well as excess reagents. And so it's essentially measuring a penalty on the chemist for what it takes to achieve a high chemical yield. Okay. Moving to E factor. So I'm going to spend some time in this presentation focusing on E factor and process mass intensity because I E factor and PMI are the most widely used metrics in the pharmaceutical industry. And I'll explain why. So E factor expands upon atom economy by focusing on measuring the total waste relative to product. I think anyone who's run a chemical reaction before has this well understood in that the amount of solvent used in a given chemical reaction and the amount of water used during an aqueous workup from a mass standpoint feeds the reagents used, the stoichiometry of reagents used, and the amount of product produced. And so E factor is intrinsically geared toward the interest of the pharmaceutical industry in that it focuses on the sheer amount of waste produced in a process. And this becomes very important when one begins to scale up your chemistry. And so on a 5-gram medicinal chemistry scale, E factor is really not much of a concern because waste is small. However, when one scales up a chemical process, waste becomes very important, and for 3 key reasons. One, when processing on an industrial scale, one must produce waste, essentially produce raw materials that essentially become waste. Number 2, one must find a way to process all of these raw materials or waste. And so this has a capacity limitation when performing chemistry on scale. And then lastly, one has to find a way to dispose of waste. And so this has cost implications, this has environmental implications, and also capacity implications. So E factor is really the best way, I think, and widely considered in the pharmaceutical industry to be the best way to capture the overall waste of a process. So this is a metric introduced by Professor Roger Sheldon in 1992. A high E factor essentially indicates there is more waste generation and with that a negative environmental impact. And so the ideal E factor is 0. I've shown here the original simple E factor that was published by Professor Sheldon in 1992, and it is nothing more than the sum of all input materials, excluding water, minus the mass of the product divided by the mass of the product. And so in other words, it's raw materials excluding water relative to the amount of product produced. As this metric has evolved, it is now widely accepted in the pharmaceutical industry to include water when calculating the metric. And so the new E factor, or complete E factor, is calculated by taking the mass of all input materials, including water, minus the product divided by the product. And one of my favorite things about E factor, I think, is that it is very simple. Is very simple to calculate, and it's very meaningful. Raws minus product divided by product is equals waste. And so this is easy to calculate for a chemist on a bench scale, and easy to calculate for an operator when working in a pilot plant. Oh, let me go back one slide. Okay, so E factor is not just limited to the pharmaceutical industry. This is actually used as a metric in petrochemicals, in the bulk chemical industry, as well as the fine chemical industry. You can see if you look at the 3rd column, E factor actually goes up. So efficiency goes down as we move down the table from petrochemicals down to the pharmaceutical industry. But this is important because, first of all, the total annual waste tonnage also goes down. As one might expect, there is much more annual waste produced in the petrochemical industry than in the fine chemical or pharmaceutical industry. And then also, as we move down the table, down the rows of the table, the complexity or value addedness of the products that are made increase. And so the complexity of pharmaceuticals is very high. They have high value in society. And so it's important to know that there is a high waste burden for the pharmaceutical industry when one thinks about efficiency as measured by E factor, which is waste, kilograms of waste to kilograms of product. However, we are producing high value added products and have an overall lower annual waste tonnage. So process mass intensity, or PMI, is essentially a close cousin of E factor. It was introduced in 2007 by the EPA and the American Chemical Society Green Chemistry Institute Pharmaceutical Roundtable as the most comprehensive metric at the time. It aimed at introducing or considering all process materials, including water and workup chemicals, as well as separation agents. And as I mentioned, E-factor has now evolved to capture all of these raw materials in its calculation. It only differs from complete E-factor by a factor of 1. This is because it does not subtract out the mass of product from the numerator in the calculation. So it's simply the mass of all input materials, including water, divided by the mass of product produced. If folks are interested, there is a free calculator, free to the public, to calculate PMI over your given chemical process or given chemical reaction. This is available on the ACS website. I've shown the link down below, in particular on the ACS Green Chemistry Institute webpage. And one can play around with this tool and measure improvements to PMI during the course of your research. And what's really nice about PMI is that the Green Chemistry Institute Pharma Roundtable has been able to capture and compile waste data from different pharmaceutical firms since 2007 and 2008. And so I've shown in the bar graph, we've been able to capture waste from a number of different firms at a number of different stages in development of a pharmaceutical product. And so as you might expect, in the preclinical phase, where focus is on speed, PMI is not very good. It's very high, but the waste burden is low because scale is small and the focus is on speed. As one considers and performs commercial process development, we're able to measure, as one might expect, a reduction in PMI, moving from preclinical to phase 1, that's where the bulk of the reduction occurs, and a continuous reduction from phase 1 all the way to phase 3, and then eventually commercial. And so this is a great tool in that it allows us to benchmark and track process waste in the pharmaceutical industry. Okay, so I'm going to transition here from the green metrics and then focus on how each one of these metrics is limited. And the bulk of the focus is I'm going to be picking on E-factor a little bit, and that's because it is the most widely used metric in the industry. But first, just focus on yield and atom economy. I think most people can probably wrap their head around how yield is limited. It is simple, but rather limited in that it does not measure reaction efficiency, and it does not incorporate solvent and water waste in the product into its calculation. And so it's simple at the mass or moles of product to the mass or moles of limiting reagent. Atom economy takes yield a step further by looking at the efficiency or waste of atoms in a given chemical reaction, but it also does not take into account mass efficiency. So it does not take into account solvent, water, and reaction yield in its calculation. And so it's not the best metric to be used in the pharmaceutical industry, because the focus in the pharma industry is, of course, on mass. Okay, and so as we move to E-factor, it is one of the key limitations, I think, in the industry is that where is one to begin starting to count process waste? And so I've shown here the commercial manufacturing process to make Viagra as published by Pfizer, starting with as aryl carboxylic acid A and the orthoethoxy aryl carboxylic acid B, and over a 7-step process calculated to have a complete E-factor of 50.3 kgs of waste per kg of product. However, if one peels back the onion, so to speak, and looks at how Pfizer manufactures fragment A, it's a multi-step sequence. And so when looking at the entire process, so to speak, capturing how fragment A is manufactured, the E-factor actually jumps up to 85.5 kgs per kg. And so roughly 30 to 40% of the overall process waste is not being captured if one is to focus just on the GMP sequence to make this product in particular. And this is certainly not a limitation of to Viagra or intrinsic to Viagra. This is a problem or a limitation of E-factor across the pharmaceutical industry. There are ambiguous and inconsistent process starting points when one calculates E-factor, and so there's a real opportunity to standardize this metric. Okay, let me go back one slide. A second limitation of E-factor is considering the complexity of the API or active pharmaceutical ingredient that's being manufactured. I think all chemists can understand that not all synthetic targets or APIs are created equal. I've shown 4 commercial APIs here on this slide, moving from left to right in order of structural and molecular complexity. So the first example is fluoxetine, manufactured by Eli Lilly, also known by the brand name Prozac. It's a relatively simple API. Then moving to the right is Viagra, as manufactured by Pfizer, where now the API contains more aryl rings, more heteroaromatic rings, and thus is of higher molecular complexity. Moving to ticagrelor, as manufactured by AstraZeneca, known by the trade name Brilinta, which is a drug used to treat heart attacks or prevent heart attacks. Now we're incorporating high number of rings, high number of heteroaromatic compounds, and now stereocenters in the molecule, which add to its overall molecular complexity. And then lastly, the last example is omasuccinate, methosuccinate, which is manufactured by Teva. And this is, on this particular scale, the highest complexity API shown. And one could easily understand, I think, that the overall amount of waste used or produced to manufacture each one of these APIs would intrinsically increase as you move from left to right. And so how can we accurately compare or measure E-factors across APIs of different complexity in order to achieve a true apples to apples comparison? Okay, and another key limitation of E-factor is, as I had mentioned previously, how is one able to compare E-factor across different APIs at different stages of development? And so one would not expect to have a comparable E-factor if one's looking at an API process used in the preclinical stage as compared to a process that had been studied for 5 to 7 years and is now launched commercially. And so as we know, E-factor improves during development and does trend downward as one moves toward commercial. However, we would like to achieve a metric that is holistic, that can capture, as I mentioned, process complexity, API complexity, but also allow for an easy comparison against our industry peers. And so a metric that can essentially measure or compare any API at any stage of development at any level of complexity. Okay, and so to summarize, how can we create a better measuring stick? How can we create a metric that normalizes process waste so that we have clear, standardized starting points, but also controls for overall molecular complexity? How can we level the playing field so that all measured E-factors are being compared, taking into account the stage or level of development? And also with the focus on, can we create a metric that is still easy to calculate? And the last point I want to mention is that this webinar is a multi-part series. And so in upcoming webinars, we will be covering a PMI predictive tool as invented by BMS to help us assist in predetermining process greenness during route selection. Okay, and so the last couple slides, very quickly, I just want to mention where the IQ Working Group on Green Chemistry has headed with respect to the limitations of E-factor. And this is where we enter the green aspiration level, otherwise known as GAL. GAL is a relatively new green chemistry metric that is the product of the IQ Working Group collaboration between 9 pharmaceutical firms and Professor Sheldon to mitigate those aforementioned shortcomings of the available process waste metrics. And so the key points about GAL is that the first and perhaps most important is that we have created consistent starting points to begin calculating E factor. And so we've established a $100 per mole rule for all starting materials used in a chemical process. And this forces firms to push back, so to speak, when calculating E factor back to true raw materials used to manufacture the API, so that all waste can be accurately captured. There are goal adjustments taking into account the complexity of a particular drug substance. And then lastly, there is a measurement tool that allows us to define greenness of any manufacturing process at any stage of development. And so this is the classic apples to apples comparison. And so this GAL actually represents the first manufacturing goal that's been introduced in terms of generous waste per kilo of produced drug. And to calculate it is relatively simple. So GAL, or the aspiration level that a firm looks to target for its commercial waste of its process, is equal to 26 kgs per kg. That's a data-derived waste constant looking at a number of different commercial processes, multiplied by the process complexity of the given API. And so process complexity equals the sum of all of the construction steps in the given chemical process. Construction steps are reaction steps, which are building skeletal CC, CX, CH, and XH bond forming steps. So bonds that are found in the final API is how we measure construction steps, and that's thus how we measure complexity. And so you take the number of construction steps in your process, you multiply it by a data-derived waste constant, which is the predicted waste per step, and you achieve the commercial target waste that you should be aspiring to, otherwise known as GAL. And so when we look at GAL overall, and we look at how we can capture greenness in using this tool, relative process greenness, to allow us to make those easy comparisons that I mentioned, it is relatively easy to calculate. And I've lined it out here in 5 easy steps. So the first step of using GAL, or relative process greenness, or RP, is to first determine the waste of your chemical process. So this is just calculating complete E factor or PMI. They can be used interchangeably here. Once you've done this, the next step is to determine the process complexity. And so that is, look at your chemical process from your key starting points at starting materials that cost $100 per mole or less, and calculate all the construction steps, or skeletal CC, CX, and XH bond forming steps in the process. From there, once you have complexity calculated, you can calculate GAL by just taking the number of construction steps multiplied by 26 kgs per kg. And then now that you have GAL calculated and your complete E factor, or PMI, calculated, you can easily calculate your relative process greenness by taking GAL and dividing it by your complete E factor. And so if you think about this calculation, you're relating in the numerator what the E factor that you should be aspiring to for your commercial process to the actual E factor of the given chemical process. And so a higher number here is better. Once you've calculated RPG, you can then compare your RPG, or relative process greenness, to the IQ team's RPG matrix, which takes into account the score that one has achieved of their RPG, and then compares it to the stage of development, and then breaks it down into 4 categories. So excellent, good, average, or below average, because I think it's pretty clear one would expect a higher RPG for a commercial process as compared to an early development process. And so in the next webinar, we will discuss in much more detail GAL, and even discuss some of the limitations of GAL and where the IQ working group on green chemistry is going to improve this metric. So in conclusion, John and I'd like to summarize that hopefully we've been able to communicate that green chemistry is a critical component of pharmaceutical manufacturing with a real and tangible impact on the planet and profit. E-factor in PMI serves as the preeminent waste metric due to its simplicity and its meaning and value. However, E-factor, while widely used, does have its limitations, and so there's a true opportunity for a new process metric. And then lastly, as I mentioned, please stay tuned. So the IQ webinar on green chemistry part 2 is coming soon, next quarter, where we will focus in particular on the details of GAL, and then also a new process metric known as iGal, as well as BMS's PMI predictor tool as new green metrics. And with that, I would just like to quickly acknowledge Mahalia for coordinating this very important webinar, as well as Ingrid for serving as our moderator and host today, and of course, John for all of his work on this particular presentation. And then I'd like to thank the entire IQ Green Chemistry working group and the IQ member companies for their support and for the opportunity to present. And with that, I think John and I would be happy to take any questions from the audience.
Okay, so I have one question. Can everybody hear me?
Yes.
Yes.
Okay, so first question. As many companies utilize external partners for everything, from development to manufacturing, how do you influence them to embrace the principles of green chemistry?
I'll take that one. There's actually a couple of approaches here. One goes along with that old saying: you can't lead a horse to you can lead a horse to water, you can't make him drink. If you give them all the tools and not try to force feed the advantages of green chemistry, but give them all the tools to evaluate what they're doing, and they see advantages in it. That's one approach. Another thing is also to relay back to the companies when you've developed or redeveloped a process that's much greener. And many times, if you're able to put that economic value in it, then they look at it and say, well, now we have an economic value. This is actually a lot wiser. We've created less waste. We don't have to pay for that. This is much cleaner, much quicker, much easier. Many times the way of treating the waste can go away based on the change in the chemistry. So as we work on it, we try to send this back to our partners to say, well, look, your chemistry was good. We found something better. Take a look at it. They don't have to necessarily look at the greenness. They look at the greenness, but they also look at their productivity. And if the productivity is increased, especially by some of the examples that we had shown today, all of a sudden their eyes open up and say, wow, we can incorporate green chemistry. In the back of their minds, they realize, well, this is an economic plus for us, a productivity enhancement, and that's what we want to do. So that's one of the approaches to take, as opposed to just trying to force feed people to embrace green chemistry. Let them realize, open up their eyes, and move forward.
Okay, thank you very much. I think we are running out of time, so I'll give it back to Maya Lair.
So we'll go ahead and end the webinar. Thank you all very much. Have a great weekend.